Hello students, welcome to the bioprocess technology lecture. In the previous lecture we already completed solid state fermentation, and next coming to the topic fermentation systems. So in this topic we are going to cover what are the different types of fermentation, like batch, fed batch and continuous fermentation system, and what are the different aspects of fermentation systems, and what are the properties of each one of it. And we are going to focus, okay, what is application based, okay, this application based principles of fermentation systems. So we can have one by one. So these are the three systems comes under fermentation, that is batch fermentation, fed batch and continuous fermentation system. The first one, the batch fermenter. So this batch fermenter is nothing but so we are going to do the fermentation process, that is the cultivation of microorganisms or animal cells or plant cells, okay, whatever may be, any biological cell cultivation. It is done, okay, with a batch process. A batch means in a time we are going to prepare the media and we are going to sterilize it, sterilize the media and pouring the media into the fermenter and maybe fitting all the components of the fermenter. Then the inoculum, the addition of inoculum to the fermenter. Then starting a fermentation process. After that, the proper monitoring of fermentation process. If the product, maybe the formation is reached to the top, okay, this optimum level, and this fermentation process is going to be stopped. So there is a graph given here. So this one, the glucose is the substrate, and the cell biomass is the cells formed, and this time, the progress of fermentation process. You can see here the maximum concentration of the substrate is going to be reduced to the bottom level, okay, maybe at the time of end of the fermentation process. And you can see the biomass is going to be reaching the top, okay, this is the stationary phase of the biomass. And after that, okay, this is going to start the death phase. So if you are extending the graph, okay, this may go to the bottom. Ultimately, okay, without any the glucose concentration. So this is the death. This is reaching the death phase. And this phase, maybe if you are plotting the values of your product, for example, if your product is an enzyme, or otherwise if your product is a biomass, or if your product is a maybe any biosurfactant, or so any valuable product of your interest. If you are plotting that also in a particular region, okay, this is the optimum level of your product. So in this point, okay, we have to stop the fermentation process, and we have to stop the fermentation process. Then we can get all the harvesting of your substances. So this is so-called batch fermentation, and the medium is added, fermenter is sterilized if it is in situ sterilizable fermenter. So that is after pouring the media, so that there can be sterilized. If it is a lab scale, and you have to prepare the media and pour the media, okay, into the fermenter and keep it, okay, within the autoclave. Okay, that is autoclavable fermenter. And inoculum, addition of inoculum and fermentation followed to completion, and finally cell harvested, and the cells are used for the further the application process. Okay, that is downstream processing. So this is batch fermenter, and the characteristics of batch fermentation system. So this is a very simplest fermenter operation, and the sterilization can be performed in a reactor. Okay, and all the nutrients are added before inoculation, and the maximum levels of carbon, nitrogen, the source, okay, are limited by inhibition of cell growth, and biomass production limited by carbon nitrogen load, and the production of toxic waste product can be limited, and the cells are harvested when biomass levels or product levels start to decline. The cells are harvested when biomass levels or product levels start to decline. Okay, maybe that is a threshold level or the optimum level of the biomass, or otherwise optimum level of the product can be considered for the stopping of batch fermentation process. So next, coming to the second part, okay, that is a fed batch. So here fed batch is nothing but this is batch 1, and this batch, so due to the maybe very long process of fermentation, that is, maybe if you want to produce antibiotics, for example, streptomycin is produced by Streptomyces species. Usually, okay, this one, all the antibiotics will be produced in the starting of the death phase of the biomass. So it will take, okay, more time. Okay, that means maybe 20 days or 21 days, maybe average days of fermentation process. If you are doing the fermentation process, okay, in a laboratory, okay, due to the sparger, and also, okay, due to the usage of the mixing, okay, that is agitation. So the exhaust pipe may lose, okay, some of the media component, okay, that is the aqueous form, that is water, can be wasted by evaporation. So this can be refilled. Okay, this level of the media can be maintained, okay, by feeding the media, okay, by a pump attached with the feedstock vessel, and this is already pre-sterilized one, and automatically, okay, this will give the sterile media, okay, into the fermenter. So this is the setup, and not only the refilling of water, and maybe if you want to get the maximum yield of your product, and you need to maintain, okay, the carbon nitrogen source. And if during the fermentation process, if the carbon and nitrogen is depleted, automatically you will get, okay, a very less number of products. So to replenish, okay, the levels of carbon nitrogen, you can also feed uh, the media components, okay, in the form of carbon nitrogen, and this can be fed into the fermenter, okay, very easily by using, okay, this type of fermenter. Okay, this is called as fed batch fermenters, and all these fed batch fermenters are very common in industries. Okay, they will fed media, or otherwise they will fed, okay, the wasted or otherwise evaporated water. So they will refill the water in case of antibiotic production. For example, okay, in antibiotic production, if you are filling, okay, maybe refilling or otherwise replenishing with the carbon nitrogen, automatically the cell division started doing it. Okay, and for the secondary metabolite production, okay, the death phase, starting of the death phase is mandatory, because at the time only for the survival, okay, this all the actinomyces, okay, started producing the antibiotics. So this is very important. Either you can refill the aqueous phase, or otherwise the media components to replenish the carbon nitrogen. So this is the type called as, okay, the fed batch fermentation process. And in this fed batch fermentation process, I know the characteristics of fed batch fermentation. And the initial medium concentration is relatively low. Okay, that is no inhibition of cell culture growth. The medium constraints is the concentrated carbon nitrogen feeds are added continuously in increments. Maybe by sequentially, okay, you can add, okay, the carbon nitrogen feeds in a concentrated manner, and controlled field results in higher biomass or product yields. Fermentation is still limited by accumulation of toxic end products. So here, okay, we are not going to remove the toxic end products, and we are not going to harvest anything, okay, during the fermentation process. So it is like batch one. So the difference between batch and fed batch, we can maybe we can understand, okay, by using the graph. So this is, we already completed, okay, this batch one, and here you can see this the biomass is also going to be increased, and the product is also going to be increased. At the same time, the glucose concentration, okay, is maintained in a constant manner. Okay, that is maybe if it is started depleting, means we can refill it. Okay, for a period of time. Okay, if it is again started depleting it, again we can refill, okay, this one, the carbon. So by this way, we can maintain the average level of the glucose concentration, and maybe the concentration, okay, can be maintained, okay, to have the products, okay, that is the biomass and product, okay, in a constant manner. So this is a very famous method, and this is very particular in the industries, okay, that is for the production of secondary metabolites, especially, okay, the antibiotic production. So I hope you understand the concept. So next one is the continuous fermentation process. The continuous means It is continuous. That means, okay, the very simple, okay, but we can represent, okay, this one is by a formula, okay, so flow rate 1 is equal to flow rate 2. Flow rate means, okay, you can pump a particular concentration or the volume, okay, into the vessel. At the same time, the same concentration, okay, can maybe you can remove or otherwise harvest from the vessel by using another pump. So pump 1 and pump 2, the capacity, the flow rate is same. That means if you are passing 1 ml, automatically 1 ml you have to take it from the vessel. So you can maintain the level of the fermentation vessel in a constant manner. That means you have to maintain the feedstock vessel, that is the sterile feedstock vessel and the collection vessel. So both should be in the sterile condition and close to environment, okay, without any contamination, possible contamination. Okay, this feedstock vessel and this pump, the pump 1 and pump 2, both are continuously synced. That means if you are passing 1 ml, automatically the 1 ml has to be recovered, okay, from the harvest pipe. So this is called as the continuous fermentation process. The main advantage of the continuous fermentation process, you can get the product continuously. At the same time, you can somehow remove the toxic end products, okay, from the fermentation vessel. In batch and that is a fed batch, so you're not going to take the toxic end products, okay, from the vessel. For example, the toxic end product means, for example, okay, if you are producing alcohol, that is ethanol production by using the yeast Saccharomyces. So we can maintain a particular concentration of ethanol within the vessel, and the ethanol, if it is increasing, okay, more than if it is reaching 35% all, okay, that will start inhibiting the growth of yeast. Okay, that may have an impact on the growth of yeast, so that may be a problem. So we need more amount of end products. At the same time, the end product may inhibit the growth of microorganisms. So in this fermenter, okay, that is continuous fermenter, you can pass the feedstock can be passed. At the same time, you can harvest the product. At the same time, you are going to remove a particular component of the toxic end product, okay, from the vessel. So it is a continuous process. You can cultivate it continuously and you can harvest the product continuously. So these are the characteristics of continuous fermentation process. The input is equal to output, that is volume is constant. The flow rate is selected to give the steady state growth rate, okay, that is the growth rate is equal to dilution rate, and the dilution rate is more than growth rate, okay, that is culture washed out. Dilution rate is lesser than the growth rate, okay, the culture overgrows. Dilution rate is equal to growth rate, means steady state culture. Okay, so these are three possibilities. Culture washout means, okay, culture goes to the dead phase. That means the dilution rate is more. That means, okay, your substrates are vanishing out, okay, from the vessel, that is from the media. That means the growth rate is dilution rate is more than the growth rate, okay, the culture washed out, and growth rate is more, the dilution rate is less, means, okay, the culture overgrows. Okay, that means, okay, you have to feed more components. At the same time, you have to maintain both dilution rate and growth rate, the steady state culture. If you are maintaining the steady state culture, you can harvest the product, okay, from the harvest pipe, and you can get a very good yield of products. And the product is harvested from the outflow stream. The stable Chemostat cultures, okay, can operate continuously for weeks or months. So minimum maybe 2 weeks to 3 weeks to 10 months, okay, we can do the continuous process of fermentation process. And this is very famous in the industry. Okay, it is not possible, practically possible, okay, to the routine, the day-to-day experiments, okay, in the academic institutions or research institutions. So we can use either batch or fed batch in the research or otherwise academic institutes. And if it is a production, for example, if you're involved in the production or otherwise research and development involved in the production, you can do, okay, this type of continuous fermentation process. And this is commercially more viable compared to fermentation process, because just you are going to feed it, feed the substrates, and you are going to get the harvest. And we can minimize the number of autoclaving, number of okay, the farm, maybe the preparation of media, and again sterilization of the vessel, and all the time, manpower. Okay, we can save a lot of things. The economics of, okay, this fermentation is much more comparable, okay, than that is more efficient, okay, when compared to batch and fed batch method. So with this, you can complete the fermentation process, and maybe if you have any doubts, please raise the questions and make a note of it, and I will answer your doubts. Okay, thank you so much.